Hello, this is Antonia,
And this is Bakung.
and we're going to be presenting our protein on alpha-synuclein. So alpha-synuclein proteins were discovered independently by at least 4 different labs before any sequence data became available in GenBank. Alpha-synuclein in its native state is an unfolded or intrinsically disordered protein. It has abnormal aggregation associated with neurodegenerative diseases. The purified form of the protein at neutral pH lacks an ordered secondary and tertiary structure. The accurate native structure of alpha-synuclein is still poorly defined since it can be intrinsically disordered, helical, or a combination of the two. The helix-rich structure has been shown to be more readily populated in the presence of phospholipid membranes, which offers one possible insight towards the functional role of the protein. Alpha-synuclein is a highly soluble presynaptic protein which is made up of 140 amino acids. In this diagram, as you can see, the first 25 amino acids are involved in a well-defined helical structure which acts like a membrane anchor to initiate docking. Residues 26 through 97 exhibits a structural flexibility and transforms between helix and random coil depending on the lipid composition. From residues 96 and on has rich of proline and has negatively charged residues, which is common in order to maintain solubility. Using electron paramagnetic resonance spectroscopy, also known as EPR and NMR, they found that alpha-synuclein was found to adopt a continuous helix structure from residues 9 through 89. The helix structure has 4 interfaces with distinct properties. The pink over here in this diagram is mainly composed of hydrophobic residues. The green interfaces over here contain several glutamate residues and is exposed to the solvent. 2 white strips down over here on the sides of the helix features 11 positively charged lysine residues, which correspond to the electrostatic interaction with lipids.
Okay, so why does or how does alpha-synuclein aggregate? Alpha-synuclein starts off as a tetramer that is thermodynamically favored inside the cells because of its high expression. The tetramers could dissolve into unfolded monomers because of its post-translational modifications, dilution, or other unknown environmental factors. Unfolded monomers can aggregate into soluble high molecular mass protofibrils and insoluble beta amyloid fibers. Pathogenic conformation of alpha-synuclein is transmitted from cell to cell, and this transmission causes disease to spread through the brain, such as PD or Alzheimer's.
Dysregulation of alpha-synuclein can lead to numerous neurodegenerative diseases, which include mainly Parkinson's, but also can include Alzheimer's. The SNCA gene, SNCA, which is the gene that provides instructions for making alpha-synuclein, has been mapped to chromosome 4 in humans. The left diagram over here shows the alpha-synuclein fibrils. In the right over here shows the cryo-EM reconstruction density map of alpha-synuclein fibrils. Fibrils are a thread-like structure that is an insoluble protein that makes up the principal structural proteins of the body. As I mentioned over here, A shows the pitch, which refers to the 360-degree helical turn. B over here shows the side view of the density map of the protein, while C shows the top view of the density map of the protein. The overall resolution of the map is 3.07 angstrom, with most side chains very visible. In this video, describes the cryo-EM reconstruction of alpha-synuclein fibrils at 3.4 angstrom resolution. It also shows the interaction between the 2 protofilaments, and it shows at 4.9 angstrom the spacing between beta strands of a single protofilament.
As you can see here, there are 3 domains in alpha-synuclein structure: the alpha lipid binding N-terminal, the amyloid binding central domain, or the NAC region, and the acidic C-terminal tail. The peptide sequence containing the N-terminal region found in residues 1 through 65. The N-terminal is positively charged, can induce alpha helical structures, and this reduces alpha-synuclein's ability to form beta structures, which is important for alpha-synuclein lipid interactions. Residues 45 and 57 is key in facilitating beta strand to beta strand interactions in the fibril conformation and acts as a hydrophobic zipper, which serves to accelerate the formation of a more mature double-stranded fibril structure. These beta structures are formed in the NAC region, or the core region of alpha-synuclein structure, and are involved in fibril formation and aggregations. This central hydrophobic domain is found in residue 65 through 95. A central hydrophobic region of the protein corresponding to residue 50, 71 to 82 was found to be essential to misfolding and aggregation of alpha-synuclein into fibrils and aggregate in isolation. Acidic C-terminal tail found in residue 96 through 140 takes on a random coil formation due to its hydrophobicity and high net negative charge due to proline. Its negative charge properties and interactions with the NAC region inhibits alpha-synuclein aggregation. Thus, when the C-terminal binds to a positive ion, the net negative charge are neutralized and the inhibitory effects disappear, leading to alpha-synuclein aggregations. Also, serine 129 phosphorylation is important as the inhibition of for inhibition, as dephosphorylation leads to formation of aggregates.
Like I mentioned before, in the STINKA gene, mutations have been found to cause Parkinson's disease. These mutations include A53T, E4K, E46K, H50Q, G51D, A53E, A53B, and A30P. The most potent mutation leading to the earliest onset of the disease is G51D. Mutations either increase in the aggregation rate or a change in the oligomeric state. Alpha-synuclein is also an acidic neuronal protein, which is found and expressed in brain tissues in high concentration and is primarily localized at the presynaptic terminals of neurons. Alpha-synuclein is expressed in hematopoietic cells and other tissues, which include the heart, skeletal muscle, pancreas, and placenta. However, the protein is more abundant in the brain.
Alpha-synuclein has multiple biological functions within the body, one of which is the regulation of glucose levels. This protein decreases secretion of insulin by interacting with insulin-containing particles, ATP-sensitive potassium channels, or KATP. This interaction inhibits insulin secretion triggered by presence of glucose and increases glucose uptake in tissue, thus reducing insulin resistance and possibility of developing type 2 diabetes. It also can act as molecular chaperone, as it contains multiple regions at C-terminal and N-terminal that are able to interact with other cellular proteins. The N-terminal domain facilitates alpha-synuclein binding with substrate proteins, resulting in the formation of large complexes. The C-terminal provides the solubilization of the large complexes. Many of neurodegenerative diseases are proteinopathies because toxic protein aggregates and fibers accumulate. This accumulation of insoluble protein aggregates with age implies a failure of cellular protein quality control system, this including molecular chaperones, the ubiquitin proteasome system, and the autophagy lysosome system. Chaperone machine failure results in inefficient neurotransmitter release, followed by gradual neurodegeneration found in Alzheimer's and PD. Over here, this shows the pathway of leucine-rich repeat kinase 2, or LRRK2, and Parkin. Parkin is a ubiquitin ligase of proteasome system that degrades redundant and disordered proteins. The most common cause for Parkinson's disease is this mutation in Parkin that affects its ligase activity efficiency, leading to insoluble aggregate formation. Alpha-synuclein interaction with Parkin in stress conditions trigger formation of alpha-tubulin deposition, altering the neuronal cytoskeleton structure and causing dysfunction. Both Parkin and LRK2 contribute to the moderating of secretory pathways and are involved in synapse development. Neuronal activity depends on regulation of synaptic vesicle dynamics, so mutation in either of these proteins could be attributed to synucleinopathy, which is a group of neurodegenerative disorders where alpha-synuclein accumulates abnormally to form inclusions in cell bodies or axons of neurons or oligodendrocytes. As you can see on the left here, Parkin mutations and LRRK mutations lead to problems. Alpha-synuclein can also accumulate in cytosols if Parkin and LRRK2 mutation affects synaptic assemblies. The overexpression of LRRK2 can destroy tubulin polymerization, influencing alpha-synuclein trafficking and lead to uneven and unwanted distribution of alpha-synuclein. This overexpression can also indirectly phosphorylate serine 129, which that we mentioned before, can cause aggregation. LRRK2 mutations are linked to faulty mitochondrial functions, imperfect biogenesis, and production of superoxides. Superoxides, or any oxidized products, can bind to a negatively charged C-terminal, triggering the formation of alpha-synuclein fibrils and aggregations, as the oxidized product neutralizes the negatively charged tail and destroys the inhibitory effect. Naturally, the malformation of both LRRK and Parkin will lead to disastrous levels of protein accumulation in neurons. Small ubiquitin modifiers undergo a process called simulation by attaching to proteins, modifying their post-translational structures, and keeping proteins soluble. It has been shown that simulated alpha-synuclein at lysine 96 and lysine 102 does not aggregate. This implies that simulation prevents accumulation of alpha-synuclein and other related diseases. Next, we discovered the pathway of alpha-synuclein and LRRK2 and its effects on other parts of the neuron systems. Alpha-synuclein is involved in modulation of neurotransmitter release, endoplasmic reticulum, and Golgi trafficking, and is loosely associated with synaptic vesicles. Alpha-synuclein interaction with tubulin in patients with Parkinson's has been linked to accelerated aggregation and fibril formation. Extracellular oligomeric alpha-synuclein has the ability to inhibit polymerization of tubulin in dopaminergic neurons, leading to apoptosis, which means cell death. This means that there's a similar interaction inside the brain between tubulin and alpha-synuclein that forms Lewy bodies, LBs for short, but on Lewy body dementia. It is proposed that environmental toxins or factors that can initiate alpha-synuclein aggregation by disrupting tubulin assembly to microtubules increases the number of free tubulins that can interact with alpha-synuclein and form fibrils. Alpha-synuclein NAC region binding to a C-terminal of dopamine transporter, or DAT, is found in patients with Parkinson's. DAT controls the reuptake of dopamine from synapse, delivering it back to the presynaptic terminal of neurosecretory cells, and any inhibition of dysfunction would lead to increased levels of dopamine, which is not a good thing. Enhanced dopamine metabolism will damage cells as it leads to oxidative stress and creating free radicals. And as we mentioned before, oxidative stress can destroy the acid and negatively charged tail that's found in the C-terminal. Normally, alpha-synuclein would be able to decrease the DAT activity and control neuronal levels of dopamine. However, aggregated or fibrillated alpha-synuclein can be neurotoxic by exposing dopaminergic cells to free radicals products of dopamine oxidative metabolism.
So many of the limitations with alpha-synuclein is understanding the folding and function of the protein, since it is very complex to the nature of the protein, since it can be either monomeric or oligomeric species. Different structures may be dependent on either the environment, definition of the native structure, post-translational modifications, and the relationship with lipids and or other agents found within the neuronal environment, which makes the interpretation of the native state very difficult. The future of this project and just the protein itself is to identify what the mechanisms of selection and translocation of alpha-synuclein into vessels are, whether or not its alpha-synuclein proteins induce neuroinflammatory responses once being secreted into the extracellular space. All these questions, more as well as the concerns about the protein, are currently being investigated and identified so that it will increase one's understanding of the basic biology of alpha-synuclein and provide effective ways to treat and diagnose people affected with Parkinson disease and many more.
Thank you for listening to our presentation. Here are the sources that we've used for this presentation. I hope you enjoy it as much as we enjoyed researching this topic. And if you guys have any questions, we're open to listen. Thank you.